So tonight we're going to be talking about gene therapy. As part of that, to introduce us to this topic, because there is some complexity here, we are going to be joined by Dr. Tim Cripe from Nationwide Children's Hospital, and he's going to give us kind of an introduction and an overview of gene therapy before we are joined by the rest of our sponsors. And just so you know who else you're going to be hearing from tonight, our panelists are going to include Dr. Louise Rodino-Klapac from Sarepta Therapeutics. We have Dr. Beth Belluscio from Pfizer. We have Dr. Fulvio Mavilio from Audentes Therapeutics, and Dr. Joel Schneider from Solid Biosciences. As a reminder, if you have questions, please submit them to the comment box below. We've already had a ton of questions come in, so I think we're going to have a great discussion after this. But to begin, we are going to start with an introduction by Dr. Cripe. So, Dr. Cripe, I'm going to hand it off to you.
Thank you, Eric. It's a pleasure to be here and be participating in this fantastic panel. I guess it's my daunting task to give a brief introduction to gene therapy, which is a large field, a complex field, a lot of nuances, and we're learning something about it every day, it seems. So I'll just give an overview to hopefully get everyone up to the same page, and maybe it'll help some of you understand what comes after this talk better in terms of the other presentations. So the first slide really shows the promise of gene therapy, and that is that you could correct a diseased cell by replacing or changing or altering expression of a protein. So a diseased cell that's missing a functioning protein in its simplest iteration can be replaced with gene therapy and result in a healthy cell. So that's really the ultimate promise. And of course, that sounds easy, but is very challenging in a whole organism and with lots of cells and different kinds of diseases to attend to. But what exactly is gene therapy? On the next slide, you can see that I've broken the definition of gene therapy down into very simple subsets. That is the use of some sort of a nucleic acid, DNA, RNA, sometimes hybrids of those two, or a mimic of those, chemical mimic, to either modify, replace, or add genes. So modifying gene expression or splicing. I showed an example of that with Duchenne muscular dystrophy with eteplirsen. Or to replace a defective gene, as there's two FDA-approved versions of replacing a defective gene, Luxturna and Zolgensma, or adding a new gene, and that's what's under trial right now with Duchenne, but there is an FDA-approved version of adding a new gene, and that's in the cancer field with something called Kymriah. Now, that's not happening in the body. It's where cells are removed from the body and they're modified, new genes added, and then they're given back to the body. So it's a little bit different paradigm. But gene therapy really encompasses all of these approaches. So on the next slide, it shows a lot of different methods of gene therapy. Many people use viruses because viruses are small particles that have evolved over thousands or millions of years to basically transfer nucleic acid into a cell. That's what they do. They're shuttles. They bring it into the cell and then they affect the cell. And there's a lot of different kinds. There's thousands and thousands of different types of viruses, many of which are under study as gene therapy vectors, which is a term we use to refer to the shuttle, the particle that brings in a nucleic acid. And so even HIV has been used for that. And in fact, that's the one that's used for that cancer application. But among those lists is a parvovirus called adeno-associated virus, or AAV, which is the most commonly used now, and particularly in the Duchenne field. And AAV, despite its name, is not an adenovirus. So the Oxford, for example, the Oxford COVID vaccine is using an animal adenovirus, but it's very different from an adeno-associated virus. They're completely different types of viruses. So if you got the COVID vaccine, you wouldn't become immune to an AAV. They're very different. But also, many people are looking at other ways to transfer nucleic acid into a cell, such as non-virus mediated. And these have been around for many years as well. They tend not to be as efficient, but technology is improving all the time. And so there are many applications that are being tested, including packaging DNA into various types of particles like lipid particles, liposomes, or condensing them onto other kinds of substrates, or even injecting them by themselves, we call naked DNA. And then there's been studies done with a gene gun where DNA or RNA are coated onto beads and shot into the tissues, believe it or not. So The next slide will describe, so that's just some of the various ways, but I'm going to focus the rest of the talk really on the use of AAV, since that's most common with the muscular dystrophies. And this slide gives you some basic principles about how the vector, the virus, gets into the cell. That does so by binding a receptor on the outside of the cell surface. And so that receptor has to match the virus or it's not going to get in there. So I kind of refer to that receptor as the front door lock. And the virus has got to have the right key. And once it gets into the cell, it's got to traverse through the cytoplasm and enter the nucleus and then deposit its nucleic acid. If it's a DNA for most viruses, some actually work in the cytoplasm, but AAV works in the nucleus. And in the nucleus, the DNA is then begins to undergo its biology. That is, it can be turned on in terms of gene expression, and it can undergo replication or copying of itself to make more viruses. In the case of most gene therapy applications, they're defective. They're not going to make more copies of themselves. They're just in there to get that gene into the nucleus, to turn it on and to affect its function. And the promoter, which is a term we use for the regulatory sequence on the DNA, that's not the actual coding sequence for a protein, but it's basically the turn-on switch, determines what cells it's expressed in and how much is expressed in that cell. So I kind of refer to the promoter as a speaker volume. So the choice of the virus serotype or the different type of virus with the key will determine which tissues it goes to, and the promoter will determine how loud or how much that gene is expressed in those tissues. Now, on the next slide, it shows that some different kinds of what we call serotypes, or those different keys, will go to different target tissues in different ways and in different amounts. So different serotypes tend to infect different kinds of tissues, but this is by no means an exhaustive table, and it just gives you a flavor that some tend to go to muscle more than others. Most of them do go to liver because they're particulates, and the liver is a big filter for the bloodstream. And they may also go to the spleen or the kidney for the same reason, sort of a non-specific uptake. But many of them will bind that lock and key in different kinds of organs. So some of these serotypes are better for nerve tissue, for example. But very few examples only go to a single tissue. I think folks are trying to engineer those now to be very specific for a different tissue. They may preferentially go to certain tissues, but most end up more than one place in the body. On the next slide, it sort of illustrates one of the principles that scientists have undertaken to try to not just rely on what nature's produced, but to actually make our own different variants of this capsid shell, this vector, this shuttle, because we can mix and match different amino acids, different proteins, and make different capsids and result in a variety of different kinds of capsid shells, and even engineer different mutations in there and select for different variants that might preferentially favor going to one tissue or another to try to increase that selectivity for a different tissue type. So if we look on the next slide in the virus itself, the wild type AAV, adeno-associated virus, meaning the wild type meaning the kind that's found naturally, has two major genes. It's got inverted terminal repeats, the ITRs, that help it package into that capsid. And then it's got two major coding genes, and actually there's 6 or so other genes, proteins that come out of these by different splicing and different promoters. But what we do in the genetic engineering world is to remove those genes. So it's no longer a replicating virus on its own. We actually provide the function of those genes that form the capsid on another plasmid or in another system, but we're able to then, what we call, gut the virus and replace the normal genes of the virus with the genes we want to get into a cell. So you can see on the bottom, the recombinant version shows the transgene in green there, which is the gene we want expressed in a cell, and then the promoter, which is that speaker volume to turn on that transgene. And there's usually some other regulatory elements, and here you can see a polyadenylation signal to get that gene to be expressed correctly. And the downside of AAV vectors is their very limited capacity, about 4.7 kilobases, 4,700 bases. Here it says 5,000. So around that area in terms of the amount you can package in that capsid shell. There are other viruses that can package more, but they also have disadvantages or advantages. Every virus has pros and cons. So this most common AAV has that limitation, which can come into play when one's considering which genes to turn on or off. Now, on the next slide, you can see one of the challenges, these are the different promoters, just examples that one can use: strong promoters that are active across many cell types, or tissue selective promoters that might usually be less strong, and they're not usually 100% specific, although we're getting better all the time at making promoters be very selective to different cells. So there's a lot of different genes, promoters in nature that one can take to selectively express a transgene in muscle or in neuron or liver, for example. And nowadays we have combinations of different sequences or synthetic promoters to try to really tune that expression better into the cell type that we want. So on the next slide, we see some of the challenges of gene therapy, and that is because we're putting a virus in and the virus isn't natural, it can be seen as foreign, and so we can have immune reactions to the AAV. And interestingly, many of us have actually been exposed to AAV, different types of AAV naturally. AAV is a virus that doesn't cause disease. It's not associated with any disease. So there are AAVs sort of circulating around the population, and many of us get exposed to those as we age, and we can have an underlying immune response to those that we don't even know about. So this immune response can pre-exist to a given AAV serotype. Now, the immune response is usually separated into two major areas, the humoral and the cellular. The humoral means the antibodies that are circulating in the bloodstream. And those antibodies can bind to the virus capsid immediately when it's being infused and prevent it from getting to the tissue that we want it to get to. But if those antibodies aren't there, or if we can do something to get rid of them temporarily, which we can in some cases, then we can get the virus to the cell of interest. As you can see here, the virus goes into the cell, as I implied before, and puts its nucleic acid into the nucleus and then produces the protein. But that can result in some other kinds of immune reactions. So for example, the proteins of the capsid shell can break down and be presented on what we call HLA class 1 on the immune system, and the immune system can recognize that and generate a cellular immune response, or T cells or NK cells, against those antigens, recognizing the cell that got the virus and killing the cell altogether. So that can be a challenge. The cell that you want to express the gene can actually be killed by the immune system. That we've gotten away without that happening in many cases now, and we've shown that doesn't always happen, but that can be a problem. Now, the capsid shell we don't make more capsid shells in these cells like a replicating virus would. So once those cells, or once those capsids are broken down, and if they're cleared, then so after probably weeks or months, there may not be any more of those foreign proteins around. But the one protein that could still be there that's foreign is the transgene, depending on the situation. So if the patient is missing a certain part of the gene, and now we're providing it, that part that was missing could be seen by that individual as foreign. And so that could also generate an immune response against the protein that's encoded by the transgene, and could also result in destruction of the cell that expresses it. So those are some of the challenges with the immune response. And not only can we lose then the expression of the transgene, but we can cause damage to the organ that transgene was supposed to be expressed in. Now, back to the pre-existing antibodies. This is a big problem in the field as well. So on the next slide, there's a lot of different studies out there, and this is just one I pulled just to give you a sense of that this isn't very rare. Adeno-associated virus is circulating amongst the human population, and many people have pre-existing antibodies. So, but it increases with age. This is one study that showed at age 2 to 7, the pre-existing antibodies against different AAV serotypes ranged from around 5 to 20% of people. And then as they age, those over 8 had more in the range of 50, 60%. And that can be a challenge. So I'm just trying to raise some of the issues that many of the speakers tonight have been able to get around in various ways. And but there are also, they continue to be challenged for us. So the next slide just summarizes some of these immunologic reactions that I talked about, just to put them forefront of your mind. The immune response to new foreign protein antigens, either the virus or the transgene, in this case, a form of dystrophin, can cause organ damage or loss of transgene expression. but not in every person. Why might it happen in some and not others? It could be relating to their tissue type or their prior exposure or other factors. And then but there are various prevention strategies that are being studied: immunosuppression, plasmapheresis, and more recently, immunoglobulin cleaving drugs to circumvent this effect. And then finally, on my last slide, I just wanted to pose some of the questions that remain in gene therapy for muscular dystrophy, some of which may be starting to be answered by the presenters that you'll hear from tonight, but many of which are still lingering and will probably vex us for a while yet to come. So what are the possible risks, and can we minimize them? Risks of complement activation and platelet consumption. We're putting foreign proteins in the body, organ damage, as we've discussed, or other kinds of side effects, putting us at risk for sepsis, et cetera. What are the benefits and how durable are they? Does each type of muscle get benefit? Is it equal or not equal? And how long does that benefit last? Who is eligible for this kind of therapy in terms of age, genetic mutation, disease status, or pre-existing immunity? And what are the best strategies to enable redosing if needed? So that just summarized, I think, to me, some of the high points of gene therapy for muscular dystrophy. And so I'm passing it back to you, Eric.
Thank you, Dr. Cripe. That was an excellent introduction. I think that was going to be really helpful for everyone watching. I do want to throw one question back to you. And I think you touched on it in the presentation, but in case any of the families missed it, we have had a lot of questions coming in about the potential of a COVID-19 vaccine being delivered through a viral vector. So maybe if you can touch on that again, on the potential impact it might have on the ability to receive gene therapy, or for those who have already received gene therapy.
Mm-hmm. Yeah, it's a great question. There are some vaccines that are being developed with adeno-associated virus. The most common COVID vaccine strategy is to take the most immunogenic or most foreign type of protein, the spike protein from the COVID virus, and express that in one of these gene therapy type platforms, so that when you get the vaccine, your body cells are making that protein, and then you're reacting against that protein. So as opposed to, some of the live virus vaccines like the live polio or the chickenpox or anything, it's not a whole virus. It's just one protein of the virus. It's actually an mRNA in most cases of the virus, so not even the DNA. So that coding sequence for that COVID protein is being expressed, and you're making an immune response to it. So it depends on which vector they're using to put it in there. So if they're using an AAV, which there are some that are being developed that way, that could be a problem for anyone trying to get AAV gene therapy. But most of the large ones are not using that. I mentioned the Oxford trial, which I think is a chimpanzee adenovirus. And again, adenovirus is not adeno-associated virus. The other companies, Pfizer and some of the others, are not using AAV to get that in. And so those shouldn't be a problem for those.
Great, thank you. I think that's going to be really helpful for those families. So we are going to move on to our next presenter. And so joining us today, we have Dr. Louise Rodino-Klapac, who from Sarepta Therapeutics. So Dr. Klapac, it's all yours.
Thank you. Thank you for inviting me to speak tonight. It's a really privileged to be here to meet with you virtually for the 6-minute talk test, and we'll see how well I do. So if you go to the next slide, my presentation will contain forward-looking statements, and that just means that I may make statements that involve risks and uncertainties, many of which are beyond our control, and you can visit our website to read these in much more detail. Today I'll be discussing our microdystrophin program. I'd like to remind you that microdystrophin is investigational, has not been FDA reviewed or approved. If we go to the next slide. Sarepta has deep experience and commitment to Duchenne with over a decade of experience. We have 14 programs in Duchenne in total, which includes 14 in RNA, 4 in gene therapy, and 2 in gene editing. Next slide. We are committed to advancing science to develop the best therapies, and to that end, we are partnering to combat key immunology challenges, as Dr. Cripe mentioned. We recognize the challenges of reaching each and every patient, but we're committed to doing our best in this case. the immune system currently limits eligibility with the presence of antibodies in some individuals. And so as we saw, typically you were using the viral vector to deliver our payload, in this case microdystrophin gene, to produce microdystrophin protein into muscle, whether that's skeletal or cardiac muscle. But when antibodies are present in some individuals, it could block the transduction or the entry of the capsid into the cell and would make this ineligible. So we're trying to combat this in multiple ways. So one, by eliminating antibodies in positive individuals. This can be done through drugs that can potentially inactivate the antibodies or remove them physically. We're also looking at ways to prevent the antibodies from forming in the first place, and that's given with the first dose of gene therapy. And finally, we're looking at novel strategies using novel AAV capsids or other ways of delivering using non-viral delivery that the immune system can't recognize for future treatment. So to summarize, we're currently conducting research with multiple partners and internally to determine how we could progress these strategies to the clinic. So next slide. So I'd like to call attention to our most recent data releases for both our microdystrophin program, otherwise known as SRP-9001, and a related form of muscular dystrophy, LGMD2E, or our SRP-9003 program. For 9001, we have two ongoing trials. The first trial, Trial 1, was an open-label study done in Duchenne muscular dystrophy boys between the ages of 4 to 7. At a dose of 2 × 10⁻⁴ vector genomes per kilogram. And we recently published our 1-year data from this trial, and I'll go into that in a bit more detail on the next slide. We also have a second trial, Trial 2, which is our ongoing placebo-controlled trial. We've enrolled 41 patients, and enrollment is complete. This is also at a dose of 2 × 10⁻⁴ vector genomes per kilogram in patients who are 4 to 7 years old. And data from this trial will be released when the study is complete. And so next, I'd just like to mention our LGMD2E trial results, which we've also presented recently. So to remind you, this trial is a open label study as well, with 2 dosing cohorts. 3 patients were enrolled at a dose of 5 × 10⁻⁻13, and 3 more patients at a high dose of 2 × 10⁻⁴, which is equivalent to our microdystrophin dose. This has important implications because this trial also used the same vector, RH74, and the same promoter, which is, you'll remember, is our light switch, which turns on the expression of the protein inside muscle cells. In this trial, patients between the ages of 4 to 13 were enrolled, and also between the weight ranges between 18 and 57 kilograms. And this is important as we gain experience with both the RH74 vector in terms of treating patients that are both older and also heavier. So we've disclosed data on 1-year safety and functional data for cohort 1, for cohort 2, day 60 safety and expression data. So if you go to the next slide, I'll go into more detail on study 1 results. We've recently published these as well as JAMA Neurology with Dr. Rendell. All right, so in terms of safety, we have seen no SAEs to date in this first study. 3 subjects had elevated liver enzymes, which resolved with steroid treatment within 1 week. And the most common adverse event that was treatment-related was vomiting, which occurred in 50% of patients shortly after AAV administration. There were no adverse immune responses or clinical sequelae associated with complement activation, and no other clinically significant laboratory findings. You can advance. We performed biopsies at both baseline and at 12 weeks. And so the first question we asked is how much of the transgene got inside muscle cells, and we found a mean of 10 to the 5th vector copies per microgram of DNA detected. And you can advance to the next. We also looked at the total amount of protein made, and this is achieved by looking at Western blot. And we see 74.3% compared to normal. If we normalize for muscle content, it's 95.8% as compared to normal. Next, we look at microdystrophin expression at the membrane. And we do this and quantify this in multiple ways. One way is counting the number of fibers, where we see 81.2% of muscle fibers expressing, and we also look at intensity, where we see 96% intensity compared to normal. And finally, if you'll advance to the following, we looked at functional outcome measures. Here I'm showing North Star Ambulatory Assessment. This is a compilation of 17 different functional measures. You can see at baseline, we saw a mean of 20.5 points on the scale across the 4 patients, whereas at 1 year, we see a mean of 26. This is a 5.5 increase from baseline, which is equivalent to about a 27% improvement from baseline, which wouldn't be predicted by natural history in this age group. These preliminary outcomes need to be confirmed. This is an open label study, and we'll confirm these in our placebo-controlled trial that's ongoing, or study 2. So if you advance to the next slide. So where are we in our development? As I mentioned, study 1 and 2 are on. We're continuing to follow patients in study 1, our open label study, and our trial 2, 41 patients placebo-controlled. All patients have been dosed, and we are dosing patients that received placebo first. Their crossover patients are now being dosed, and we'll release those data when the trial is complete. Finally, we'll be beginning our late-stage studies in the second half of 2020. This is a group of studies referred to Study 3, with the goal of expanding eligibility criteria across both mutations and ages. The first of these trials, of the trial 3 trials, will be in the same age criteria, similar to trials 1 and 2. And we are also currently planning a trial which will involve individuals who are non-ambulatory. These studies will include commercial process material and will support global regulatory filings and access and reimbursement. So if you go to the last slide. So thank you so much for having me speak today. This 6-minute talk was difficult, I'll tell you, but I appreciate it. And if you have any questions, you can always reach us at advocacy@sarepta.com. So thank you, and Eric, I'll turn it back to you.
Thank you, Dr. Klapac. And we have a question for you. So excited to hear, the news of some of the partnerships going on. Can you give us maybe any timeline for when we might expect to see some of those in the clinic? I know there's always a lot of excitement and interest about how we can get around pre-existing antibodies and tackling challenges like redosing. Sure.
Yes, we're definitely working as fast as we can with those partnerships. We're performing non-clinical studies right now, research studies, to be able to prove that they're effective at either removing antibodies or preventing those from forming. And so we're in early stages, but we're moving as fast as we can. So.
Great. Thank you. And we're going to go to our next speaker now, which is Dr. Beth Belluscio from Pfizer. So if you're ready, Dr. Belluscio, I'll pass it over to you.
Great. Thanks, Eric. Thanks so much. It's really a thrill to be part of this phenomenal conference that PPMD is putting on, and I'm really grateful to be included in this panel on gene therapy. So I'm Beth Belluscio. I'm the Global Clinical Lead for Rare Neurological Disorders at Pfizer. And if you go to the next slide, I have a very similar slide to Dr. Rodino-Klapac about forward-looking statements. So then we can go on to the next slide, where I'm really pleased to report about our ongoing phase 1b study. We continue to enroll study participants, and in fact, this slide is already a little bit out of date. We now have dosed 15 boys, 3 at the low dose and 12 at the high dose, and we'll continue to enroll additional participants. Just to update everybody a little bit on the status, with regard to safety, we have had a total of 3 serious adverse events. 2 of those Michael Binks presented about at the PPMD conference last year, and then the third he presented at a conference earlier this year in May, and we've had none since that time. At that conference in May, Michael Binks also provided a thorough review of our preliminary signs of efficacy that we think are very encouraging. And that full presentation is available at Pfizer.com. So I'm not going to review all those results here. But the main thing that's really important about the results of our phase 1b study is that they're supportive of us moving into our phase 3 that we're really excited about. So on the next slide, I want to talk a little bit about our Cifrio phase 3 study, which is a randomized, double-blind, placebo-controlled study, as I think most people know. It is currently listed on clinicaltrials.gov, and so all the details of the study design are listed there. I've already also given a couple of presentations recently to describe some of those details of the study design, and those are available on the internet, including one that's a YouTube video, so it's kind of easy to find. But just to remind people, it is a global multi-center trial, and we are actively moving forward to start the study as fast as we can. We haven't paused our program at all due to COVID, although as people know, I think there's a really tremendous strain on the healthcare system right now, and that is having some effect on our sites and on their readiness and on some other factors that we don't have any control over. But the good news is we're still on track to start the study later on this year, subject to regulatory approval. So as I mentioned, this is a global phase 3 study, and we have multiple sites, as described a little bit more on the next slide. In the United States, we have many, many sites planned, and as illustrated in this map, we have at least one site, usually many, within every region of the United States. And really our aim here is to make sure that patients and families who want to participate in the study won't have to travel very far to a site. Now, each site, as it's really ready to and open to enroll, will be posted on ct.gov. So that's the place to look for sites that are opening, and one should be near everybody who's interested in participating. We also will have multiple sites outside of the U.S., and those will also be posted on the ct.gov site. Now, in addition to this phase 3 study, the Cifrio study of in the ambulatory population, as I think many people know, we also are planning a global multi-center randomized control trial, phase 3 study of the non-ambulatory population. And before that study starts, we are planning to include a few non-ambulatory subjects in our ongoing phase 1 study. The numbers will be fairly small, so as investigators screen patients for enrollment, they'll probably choose those subjects mostly from their own clinics. But we're really looking forward to our much larger study that will have opportunities for lots of non-ambulatory patients. So now I kind of want to turn to a different topic on the next slide. And this is around seroconversion. That's kind of a big word, but really all it means is that if you go and you test for neutralizing antibodies and you're negative, that sometime in the future, you know, a year or two later, if you've been exposed to AAV, you may have then developed neutralizing antibodies and you'll be positive. So going from negative to positive is what seroconversion means. And some people have been wondering whether that can happen if you've been around someone who's been treated with gene therapy. And this is sort of illustrated in the little cartoon on the right, where the boy in yellow is receiving his gene therapy. And then soon after that time, some of those AAV viral particles could be present in some of his bodily fluids, like his saliva. And then let's say his brother in the red shirt is nearby and comes into contact with his saliva, then that boy in the red shirt may develop neutralizing antibodies. We don't think that would make him sick in any way, but if he were someone who were interested in gene therapy in the future, then that might affect his chances. Now, again, we don't really know if this happens, but it's really important, I think, for us to understand if it happens or not. So at Pfizer, we're going to do a couple of things to try and find out. And if you go to the next slide, really a key part of our plan is something we're calling the household contact study. So this is a whole separate study, but it's sort of a sub-study to our phase 3 Cifrio trial. In the sub-study, what we'll be doing is asking household members, family members of the Cifrio participants if they'd be willing to volunteer. And what all they need to do is have 3 blood draws in their home, and we'll be testing their blood for neutralizing antibodies. And this way, when that study concludes, we'll be able to have actual information on whether or not people do have seroconversion when they're around somebody who's been treated with gene therapy. So I think this is really important for the entire community, and we're looking forward to being able to share that data. Now, until we get those data, we will be looking to take care of and help the siblings of boys who are treated. And the next slide describes a couple things we'll be doing along those lines. The most important thing is, in our Cifrio study, if there are 2 boys who both qualify for the study, they meet the inclusion-exclusion criteria, we'll be randomizing them to the same cohort. And that eliminates the chance that one boy could get the investigational gene therapy and then cause the other to develop antibodies before he can get treated. That really wraps up what I wanted to talk about today. If you go to the last slide, I just wanted to really express our gratitude to all of our partners who have given us a lot of input on our trial design by letting us know what's really important to patients, and that really helps us be able to bring therapies that are important to this community forward. So thanks a lot. Back over to you, Eric.
Thank you, Dr. Belluscio. I think we're all going to be really excited to see the results of that seroconversion study that's asking a really important question that we need to address. I was wondering, though, if you could expand a little bit on, in the slides it mentioned, in response to the serious adverse events that you had previously seen, there were some additional safety measures. Could you maybe expand on what you are doing to help mitigate those risks?
Absolutely. So really the most important thing we've done is to learn how to anticipate what's going on with these patients. So there seems to be a pretty set pattern of the response, and we can, and therefore observe it and monitor for it very carefully. So we have patients stay near the site for a couple of weeks. We can monitor their labs very closely, and that way we can really be on top of anything that could be happening and treat it effectively. And we're really hopeful that is going to be good in the future.
Thank you. And with that, we are going to go on to our next presenter, Dr. Fulvio Mavilio from Audentes Therapeutics. So I'm going to pass it to you, Dr. Mavilio.
Good evening, everyone. And thank you for inviting me to this extremely interesting symposium. In the next slide, this is the usual disclaimer slide. I think we can skip it in the interest of time and go to my real first slide. Audentes is a biotechnology company, the mission of which is to develop genetic medicines for devastating diseases with the potential to be transformative, and particularly for neuromuscular diseases. Audentes was recently acquired by Astellas, a multinational pharmaceutical company. So now we are a division of Astellas and a so-called center of excellence for genetic medicines. In the next slide is our pipeline. As I said, we are focused on neuromuscular diseases. We have in advanced clinical development a gene therapy, an AAV gene therapy for X-linked myotubular myopathy, a second gene therapy product for Pompe disease, which is at the beginning of its clinical phase, and the first 3 products for Duchenne muscular dystrophy. I will get into more details of this, but these are gene therapy products aimed at skipping exons, and in particular exon 2 or exon 55 or exon 53, and finally another product for myotonic dystrophy. So in the next slide, what we are doing for Duchenne. At this point, after yesterday's session and after great Dr. Cripe's introduction, you know everything about AAV and you know everything about exon skipping. So what is that we are doing? We are essentially using exon skipping, which is pretty much what the antisense oligonucleotides that are developed by several companies, and you know about this, are doing to skip exons to restore reading frames in many mutations of Duchenne muscular dystrophy and allow for the production of a functional dystrophin product. So we are doing exon skipping, but what is different is the modality of delivering this interesting molecules. What we are using is a gene therapy vector, is an AAV that carries a gene like all AAV vectors, but this gene doesn't produce a protein like the microdystrophin you've just heard about, but rather a small nuclear RNA, which is the hairpin structure that you see in the right part of the cartoon, that carries this little red bar, which is an antisense sequence, very similar to what you know for the antisense oligonucleotides, except that this is not an oligonucleotide. It's an RNA sequence which is part of a larger RNA, which is the structure you see in this cartoon. So essentially, we are using antisense to skip exon and reconstitute reading frames, but with a different modality that we believe is more efficacious. Why? First, because we use a vector that puts this molecule, this effector molecule, into the majority of fibers in the muscle and in the heart, which is very important. Second, because this antisense is an RNA molecule, and the splicing machinery normally into cells is based on RNAs and not on DNAs. Third, because this small nuclear RNA, this hairpin structure you see here, is actually a modified RNA, which is normally part of the splicing machinery. So we are actually targeting the antisense in the nucleus of the cells where the action is, and when we think the skipping is going to be more efficient. Now, preclinical models in animal models of DMD with that mimic mutations that are typical of patients, we have observed excellent delivery of these molecules to the majority of muscle fibers and to the majority of the heart fibers. We have seen exon skipping up to 80% of the RNA transcript that comes from the dystrophin gene, and robust synthesis of functional dystrophin. So based on this type of preclinical studies, in the next slide, the team of, before going to that, what's the result of this skipping? Well, you know the game at this point. Dystrophin is a huge molecule, and all those colored boxes give you a sense of how many domains, functional domains, are present in the dystrophin. So normal human dystrophin has this different domains of different colors, an amino terminal in red and a carboxy terminal in purple, and other domains in the middle. In making microdystrophin, we have to remove some of these domains, so have a protein that is missing some of its important function. Exon skipping, no matter what modality you use, essentially gives rise to a larger molecule, which carries an internal deletion that depends on the patient's mutation, of course, but it's essentially much larger than a microdystrophin and contains most of its functional domain, in particular the carboxy terminus, that's very important, the NOS, so-called binding domain, which is also very important, and of course, the length, which is important for the mechanical properties of the molecule. So as I said, based on preclinical studies done with these modalities of exon skipping, in the next slide, the team of Kevin Flanagan and Megan Waldrop is the principal investigators of this study, is bringing this to the clinics. So this is a first in human. It's a proof of concept study for patients that carry a duplication of exon 2. So the skipping here is removing one or both of these duplicated exons. 2 patients have been already treated in this trial with a starting dose of 3 × 10^13 vector genome per kilogram. The patients have been dosed right before the COVID-19 crisis, but unfortunately COVID-19 delayed a little bit the analysis of these patients at the first time point. and Megan and Kevin will report on the result of the first biopsies as soon as they are ready to do that. This study is done in collaboration with Audentes, with collaborators with this. We hope to capitalize from the knowledge that this pilot study will give us in terms of efficiency of skipping, efficacy of producing dystrophin, and possibly ameliorating functional parameters in patients. And based on that study, we will fine-tune the development and construction of our vectors that, in the last slide, are essentially aimed at skipping of many different exons. Here in this pie, you essentially see the proportion of patients that carry mutation that can be corrected by skipping one or the other exons. For instance, 13% of patients are amenable for exon 51 skipping, 8% for exon 53 skipping, and so on and so forth. So with 6 to 8 products, we believe we can address more than 50% of the prevalent patient population. And of course, developing 5 or 8 different products is 5 or 8 more times more difficult than developing one. So the way we want to do this, and we may want to we can address this in the discussion if we have time, is through a platform approach. So essentially developing a series of vectors that would be absolutely identical in everything except for the small antisense sequence, which will, of course, will be specific for each mutation. This work is ongoing, and as I said, essentially for duplication of exon 2 and for exon 51 and 53, which are of course, the most frequent prevalent population in humans. And with that, I will stop by thanking you for your attention and the PPMD for giving us the opportunity to discuss this, that as you can see is at the very beginning, this journey into gene therapy, exon skipping gene therapy. Thank you. Back to you, Eric.
Thank you, Dr. Mavilio. We're always excited to see new strategies enter the Duchenne space. Now, we do know that many in the DMD community have heard about the 2 deaths that occurred in the X-linked myotubular myopathy gene therapy trial. We were hoping you could share with the DMD community a summary of that situation and what is known at this time.
Sure. Well, first of all, we're very saddened to share the news that 2 patients unfortunately passed away in our MTM gene therapy trial. And of course, our thought as a company and as scientists and members of the rare disease community go to these families in a very, very difficult moment. So what happened? As I said, 2 patients passed away. And of course, we are committed to be as transparent as possible and give the patients community, not only the MTM patient community, all the information that are necessary to understand what happens. First of all, we have treated 23 patients in the MTM gene therapy trial. 6 patients at the lower dose of 10 to the 14 vector genome per kilogram, and the rest at the higher dose of 3 times 10 to the 14 vector genome per kilogram. The patients that unfortunately died were part of this second cohort of patients, so they were treated at the higher dose. But that's not the whole story. So the immediate cause of death for both patients was sepsis, which means generalized infections that could not be, unfortunately, controlled and eventually caused their death. But this came at the end of a pretty long period of hospitalization due to liver conditions, to what became severe liver dysfunction, and actually started 4 weeks after dosing as elevated bilirubin levels. This is something that we have not observed before. As it turned out, a significant fraction of the MTM patients do carry a liver condition that is a mild fluctuating bilirubin levels, and this appears to be part of the disease. It was probably overlooked in previous natural history studies. But in the natural history study that we carried out before and in parallel to the interventional studies, we did observe that a significant number of patients carry these conditions. So these patients were, the 2 patients that died were older than the average patients in the trial. They were more than 5 years of age. They were heavier. And since the dose is adjusted to weight, as you know, they also received the highest total dose of vectors. So at the moment, of course, the investigation is ongoing. We don't have any definitive conclusion to propose to you. But what we believe is that it is a combination of factors, but the pre-existing liver fragility we can define is probably an important component of this for a vector like the AAV that massively goes to the liver. It could induce liver toxicity that is in general well tolerated or controlled. In these patients could be in fact devastating. We are discussing with the agency, of course, trying to introduce corrective measures in our therapy in order to reduce the risk for these patients. I have to say that other patients that were younger and smaller with similar pre-existing liver conditions that were treated at the lower dose did not develop any serious adverse event. So we are learning and we are working with the agency in order to reduce the risk for this patient. I just want to conclude by saying that MTM and Duchenne are very different diseases. MTM is a devastating, lethal disorders in the first 2 years of life. So what we are seeing here and what we are learning is something that would be important for us to learn also for our future gene therapy trial for Duchenne. But you have to consider that the risk for these 2 populations is very, very different.
Thank you. We know that's a really difficult subject to discuss, but we do appreciate the transparency, and I think everyone in the Duchenne community appreciates that as well. And so we're going to move to our next speaker. We have Dr. Joel Schneider from Solid joining us. So, Dr. Schneider.
Thank you, Eric. Good evening. My name is Joel Schneider, and I'm the Chief Technology Officer at Solid Biosciences. I'd like to thank PPMD for hosting the event this evening. And tonight I'd like to give an update on our Ignite DMD clinical trial, testing a microdystrophin, as well as a bit of an update on a press release, which we put out immediately before the panel this evening. Next slide, please. These are my forward-looking statements, like the others. Next slide, please. As a reminder, Solid Biosciences was founded in 2013 by families directly affected by Duchenne muscular dystrophy. Since then, we've been developing different therapies for Duchenne, including our microdystrophin gene therapy candidate called SGT-001. As others have described, the candidate is designed to replace the underlying deficit associated with Duchenne, which is the absence of a protein called dystrophin. Next slide, please. SGT-001 contains a differentiated microdystrophin construct, and it's based on 30 years of research by our advisors, Dongxing Zhuan and Jeff Chamberlain, and it shows robust protein expression as well as the unique restoration of a protein called nNOS, which Fulvio mentioned before. The image on the right highlights data from one of our clinical biopsies taken 3 months after administration in our 2 × 10^14 vector genomes per kilogram cohort, showing the expression of a microdystrophin alongside the restoration of functional nitric oxide production in the muscle bed. Next slide, please. Ignite DMD is our phase 1/2 clinical trial, which is currently on hold. And to date, we've dosed 6 patients in the trial, 3 at the dose of 5 × 10^13 vector genomes per kilogram, and 3 at the dose of 2 × 10^14 vector genomes per kilogram. We have 2 primary endpoints, safety, of course, as well as SGT-001 microdystrophin expression 12 months after administration. We also have a number of secondary endpoints associated with the trial that allow us to better understand efficacy and functionality of our microdystrophins. As a reminder, SGT-001 and Ignite DMD are currently on hold because of observations of serious adverse events in the last patient dosed. And I'll talk a little bit more about that in just a moment. But importantly, as a reminder, all patients within the trial are doing well, including the last patient dosed. Next slide, please. We're encouraged by the expression that we've seen to date. And SGT-001 dosed at the 2 × 10^14 vector genomes per kilogram dose leads to robust, widespread expression across the muscle bed, as highlighted on this slide. Next slide, please. So where we are today, we have, as I highlighted in the press release that came out just before this gene therapy panel, we heard back from the FDA today that the clinical hold on IGNITE DMD will continue in place. And they specifically responded with requests for further manufacturing information related to process improvements that we're making in the SGT-001 production process. They've also requested updated safety and efficacy data for all the patients dosed, and they provided additional directional information on the total viral load to be administered per patient. Our clear next steps and immediate goals are to engage with the FDA to determine the path forward for the program. And we're encouraged by the microdystrophin expression that we've demonstrated to date. We continue to follow the current study subjects per protocol, and we're looking forward to evaluating samples and functional endpoints taken from 12-month time points. These, of course, are being done with consideration of the current COVID-19 environment. I understand that there are many questions related to our hold and our next steps, and we'll share updates regarding IGNITE DMD with the community in the coming months. Next slide, please. We're grateful to the community for your continued support and the interest in our program. And together, we're going to continue in our relentless pursuit to improve the lives of patients with DMD. Our sincerest thanks for PPMD for quickly adapting this trial, even regardless of the COVID-19 pandemic, and for giving Solid the opportunity to be part of this really intriguing panel. Thank you for your time, and back to you, Eric.
Thank you, Dr. Schneider. And thank you as well for sharing the comments that you received already from the FDA. I know that's something as pretty quick turnaround time, but again, we appreciate the transparency. I actually wanted to follow up on something from your construct. We have heard about the nNOS binding domain before. Could you maybe clarify for the community what you see as the significance of that, and just kind of dive into that a little bit?
Sure. As Fulvio highlighted, normal dystrophin expression is correlated to the expression of a number of dystrophin-associated proteins that play various roles in normal muscle function. Nitric oxide is produced in a healthy muscle during periods of energetic need. And so when we exercise and the muscles require more oxygenation, nitric oxide is produced, oxygen is supplied to the muscle beds, and that allows us to respond to energetic needs. Duchenne patients lack dystrophin and therefore lack the restoration and localization of these other critically associated proteins and therefore lack nitric oxide. And in preclinical models, we've been able to demonstrate that the restoration of microdystrophin alongside the association and restoration of normal nitric oxide production leads to various differentiated functional improvements. We were excited to see in our patients dosed with SGT-001 in the 2E14 cohort not only the expression of microdystrophin across the muscle bed, but also functional nitric oxide production in the muscle as well. We also saw the localization and expression of the nitric oxide synthase protein as well. Now, we've got a lot more in front of us, and today we're really focused on removing the hold and working with the FDA as closely as we can. We are excited by the data we've seen so far and the unique differentiation in our microdystrophin construct.
Thank you. And we're going to open it up now to our full panel for a Q&A. So again, if you have questions, please submit them below. I think we are going to just kind of cancel our last polling for tonight. We have a lot of questions. We're not going to be able to get to all of them tonight, but we will pass those along to the sponsors. And just as a first one, Dr. Cripe, I'm actually going to throw this to you. We've had a lot of questions come in now that we see different AAV serotypes, RH74, AAV9, AAV8, and a lot of patients and family members are asking, if I test and have pre-existing antibodies to one of these vectors, is it possible that I could still receive gene therapy from another one?
Oh, that's a great question. A lot of the antibodies do cross-react. So even technically, the word serotype originally meant that you only reacted to that type and not to other types. a lot of the AAVs do share a lot of common sequences or epitopes, which is the term we use for what an antibody recognizes. And so many of the antibodies that you might generate for one virus will cross-react to other AAVs. So the only way to know is to get the eligibility testing for any given study where they test for antibodies for their particular vector.
Thank you. I think, Ryan, you had a question?
Yeah, so, and this is really for all, and we'll probably just start around the circle. These are early days, and families are having to make decisions. And I guess the big question for so many is the uncertainty around durability. So you're making a choice for a gene therapy, and it's a one shot for now. We're all talking about redosing. I think we can admit it's not around the corner. So I just wanted your general thoughts on this sort of first generation for Duchenne gene therapies about durability based on preclinical and even based on the early results we're seeing. So Joel, I'm going to pick on you because you are on the top right of the square there.
It's a little like the Brady Bunch.
It totally is.
It's a great question, Ryan. And enrolling in a gene therapy clinical trial is a personal and difficult decision that, of course, should be done in consultation with your physician. It is early. This field is very nascent. But I think in general, the data that's been generated so far is certainly promising and encouraging. The best data that we've generated from our preclinical models is in our dog work, our dystrophic dog studies, where we've gone out for biopsies serially taken over various points of time and seen over 3 years of expression. Now, a dog isn't a human, and a mouse isn't a dog, and a mouse certainly isn't a human. And so we have a lot more to understand as we all assess durability. Louise highlighted their 1-year follow-up, which is, of course, also very encouraging. And muscle does seem to be uniquely suited for maintaining transgene, which is the gene that we're delivering, maintaining durability alongside the expression. And it may be because muscle tends to be a fairly slow tissue to turn over.
Okay. Louise?
I would agree. And, durability is a tough thing to assess because there's no way to speed up time, right? So we continue to gain this information over time and relying on non-clinical models and clinical data. So we're encouraged by the clinical data that we have so far in our microdystrophin program up to 1 year, and then also in the limb-girdle program as well, where we didn't see any drop in function over that time. We saw sustained functional improvement. And so together with non-clinical data, we have mouse data for the lifespan of the mouse. There's data from the golden retriever muscular dystrophy dog for at least 6 years. So I think as the field's collectively collecting this data, I think we're encouraged about the durability of treatment, but there'll be many years of follow-up and continued study to be able to anticipate the total outcome.
And Beth, I see you shaking your head, and imagine you you agree with those statements. Anything to add to that?
No, I think we have sort of similar data that aligns with what people have said. I'd say the only other thing is that really the most important thing about durability is how patients will do, right? How they'll function. And so I think all of us probably have trials that are going out for a prolonged period. I know our trials are all at least 5 years long, so that over time we will be able to track, how patients really function and whether that's maintained over the long period.
Yeah, Fulvio.
I can give you 2 pieces of data. The first is for myotubular myopathy, we carried out for years studies in the dog model. The earliest dogs that were dosed are now 9 years of age, and they're still doing fine. Actually, they became the pet dogs of the investigators. So in a model which is not a dystrophy, so it's a different disease, which like myotubular myopathy, at least in the dogs, we have a duration that can go up to 9, possibly 10 years. Mm-hmm. The second piece of evidence comes from studies that I carried out in my previous life as an academic investigator on the dog model of Duchenne. And what we found is that what is very important in order to durability to be long is the number of fibers that you can actually transduce with your vector. What we found out is that if we transduce, or if we put our product that in those studies was microdystrophin in less than 50% of the fibers, regeneration, degeneration will continue to go on, and then you will essentially lose your vector much more quickly than if you transduce more than 50% of the fibers. And if you transduce more than 80% of the fibers, that stabilizes the degeneration-regeneration cycle and essentially grants a much more durable effect. I think that this is important, and I was very pleased to learn from your presentations that in all these clinical trials, the number of fibers that are transduced with microdystrophin are actually very high. That's an important issue, and it's important when we think about dose escalating. It's difficult to start with doses that we can predict are not going to allow for a durable effect. And that's part of the reason why we all tend to start with doses that are relatively high.
Thank you. Just to follow on to that, as we get into the older non-ambulatory patients with less healthy muscle tissue, the risk certainly may be going up then if these doses, these are high doses of virus. I hate to kick it around the room again, but this is a population, we're really looking at, we want more treatments for, but what does that look like in terms of the risk benefit as patients get older and there's less healthy muscle to essentially play with, for lack of a better phrase, or treat? We're going to go around the room again, and we're going to start with Joel.
Sure. Thanks, Ryan. I think moving forward in general in this field, we're taking the iterative, stepwise, cautionary steps with the ambition of ultimately, hopefully, dosing all patients with Duchenne. Again, from the preclinical world, there's data that's been generated in older animal models that suggests even with decreased amount of muscle mass and increased inflammation and fibrotic presence, certainly animal models can, of course, still benefit. Solid's first patient dose was a non-ambulant patient. And so our ambition is certainly to go back there. Today, of course, as I highlighted, we have to work to have the hold lifted. But I think the overall approach that you're hearing about is really humble in the sense that we're all taking safe, cautionary steps forward, trying to understand the body's response to these major infusions of viral particles, and based on the knowledge that we're generating in the clinical experience, making the responsible steps forward.
Mm-hmm. Yeah, and in the interest of time, unless somebody has a strong disagreement, I want to let Eric ask another question, or if someone wanted to add anything to that, please do.
All right, great. So we've had a number of questions come in from different families asking about specific mutations, whether it be duplications, nonsense mutations. And again, we're going to go around to everyone, and maybe you could expand on, in either your current trial or upcoming trials, any restrictions that you have around specific mutations. And Fulvio, I'll actually start with you on this one.
Well, of course, exon skipping is by definition mutation specific. So for each one of our product, we will enroll patients that have mutations that can be corrected by skipping one particular exon. So again, by definition, the type of product that we are developing are not addressing the whole patient population, are actually addressing a fraction of the population for each specific product. We hope to be able, through the platform approach, to have products that will present the same toxicity, the same risk, the same efficacy in terms of delivery and exon skipping, and target each specific mutation. But clearly, with our approach, we cannot possibly address all the mutations.
Great, thank you. Beth?
Right. For our AAV gene therapy, exclusion at all for underlying mutation.
I'm sorry, could you repeat that? It was a little bit choppy coming through.
Yeah, okay, good. Short answer, we don't have any exclusions for underlying mutation.
Great.
In terms of mutation, you just have to have one that corresponds to Duchenne.
Thank you. Louise?
So our first two trials, we did have an exclusion for mutations between 18 and 58 of the dystrophin gene, but for our future trials, we will not have those same restrictions on mutations.
Great. And Joel?
We have, can you hear me?
Yes.
We have no exclusion criteria related to underlying mutations, just a diagnosis of DMD.
Great. Thank you. This one came in for Audentes. Do you have a timeline for when you'll have your IND submission for your phase 1 trial?
We are developing our timeline. As I said, we are essentially collaborating with Nationwide on a first proof of concept trial. Our timeline in terms of Audentes specific products will largely depend on the preclinical studies, the discussions that we're having with the agency, the result of the proof of concept studies in terms of dose, efficacy, and toxicity, of course, and safety. We don't have a real timeline yet, but we are confident that.
Thank you.
I heard he's confident.
I think that might have been on our end. So we have another kind of broad question that we've had come in a number of different times, and it's questions about steroids, both leading into the study and kind of coming out after receiving gene therapy. So knowing that different families are on weekend dosing versus daily dosing, prednisone, deflazacort, are there any restrictions or that for families as they're looking to join trials that they should be considering, either kind of preparing for a trial or kind of after the delivery phase? And so I'll start with you, Louise.
Sure. So if our current trial, patients are on daily steroids for the first 30 to 60 days for delivery. For our next trials, we'll continue a similar paradigm, and we'll, as we release the design of our next trial on clinicaltrials.gov, we'll sort of go into a lot more detail on the specifics of our future protocols for the next studies.
Thank you. Beth, could you share what is planned for the CIFRO study?
Yeah, absolutely. So our patients need to be on a daily dose of steroids coming into the trial. We have specific minimums that it's hard to remember them if I just say them out loud, but they are listed on our ct.gov site in terms of the minimum doses, because it's a little different for prednisone versus deflazacort. We do then increase the steroids slightly right after dosing, and we found that to be very effective in minimizing some of those immune responses. And then the patients taper back down to their background, their dose after that.
Great, thank you. And Joel, you as well.
Very similar design to Pfizer, where for 6 months before administration, patients need to maintain a steady course of their standard of care. The dose is slightly elevated before administration, and then subsequent to administration is tapered back down to their standard of care over a period of time. Great,
thank you. And I just want to take this time to thank all of the participants today in our gene therapy panel. We really appreciate you taking the time to be with us today. Dr. Cripe, again, thank you for that wonderful introduction. But we had a lot of questions that we weren't able to get to. We do apologize for that. We will make sure that we get those to the sponsors. But we wanted to, again, thank you all.
Thank you, everybody.
Thank you.